Kumaralingam. So
Yeah, ma'am.
please, are you there, sir?
Yeah.
Yeah, sure. All right, you can start your presentation, please.
Yeah, ma'am. Audible? Hello?
Yes.
Yeah, ma'am. Good morning, ma'am. I am Dr. Kumaralingam, working as a scientist B, Sathyabama Institute of Science and Technology. And first of all, I thank to emerging scientist teams. They selecting me to given a good opportunity to me in front of your audience. I am going to present my conference entitled Food and Feeding of Selective Species of Brachyuran Crabs from Andaman Nicobar Islands. Crabs occupy many different niches and inhabit in a variety of geographical areas. The variety of food consumed also. There are several reports on food and feeding of various species like Silas areata. In the present study, to investigate the habitat ecology of food and feeding habitats of Andaman Nicobar Islands. Methods. Data collection was made from 2013 to 2014. Two selective sites were chosen to collect the samples, particularly at Karmatang Beach in North Andaman, coming under Andaman, and Sastri Nagar in Great Nicobar Islands. I will show that plate 1 actually. All the collected crabs were immediately preserved in an ice chest with a later transfer to -20 in the laboratory condition period.
Sorry, Dr. Kumar, are you moving your slide?
Yeah.
Because is it mine or all? Because your slide is static for me.
It's not showing, ma'am.
Yeah, it's not moving.
Now moving, no?
Yes. Can you put in the slide mode, slideshow mode?
Yeah, I'll put the slideshow mode. Now you are showing.
We can, yeah, we can see your slide, but yeah.
Now okay, na, ma'am. Shall I proceed?
Yeah, sure, proceed.
Yeah. A total number of 160 specimens of Brachyuran crabs were examined from the food analysis the cardiac stomach specimen. Dissected out of the content fixed in the 5 to 10% of formalin. The stomach content examined under the stereo microscope, 40x magnification, where the ideally dissected to order and until the possible taxa. The alimentary items were identified the methods for the frequency of occurrence, Williams 1981. The showing the plate 1, A, food and feeding analysis of Brachyuran crabs in Karmatang beach area, in particular in North Andaman. The species and zoea is stomach content of prey items. The D is showing that Sastri Nagar of Great Nicobar. The species name Leona Fugli. The stomach content was analysis for the cardiac. In the result, a total of 160 stomach were analyzed and the grouped in major categories of crustacean, polychaetes, fish larvae, nematodes, bivalve velinger, gastropod velinger, zooplankton, phytoplankton, and unidentified food items. Wherever the food was found in any stomach, in always a content mixture of various food groups. The analysis it was found that percentage frequency of occurrence, crustacean is a major contribution of that in the prey items, 80%, and followed by polychaetes, algae, and unidentified prey items is a 50% of, were recorded in the two species of Paracan panelism and Lulina fugili. The major food items in crab stomach were observed in the crustacean fragments. The plate shows showing that major prey items observed in the Ketoneta, copypod, larvaceae, and polychaete larvae. The discussion. In the present study, reef associated 8 species in Brachyuran crab are found in the two stations. I will show you the two species only. In Karmatang Beach and Sastri Nagar, maximum species are found over there. The diet of coral reef associated crab found to be the several aspects of regarding the diet and other crabs. Also, was observed in the study which is a present Persena and Zoocyminus, Grappulus, Albinotus, Philomenus. Those the species are most favorite diet food of crustacean, polychaete, and phytoplankton and zooplankton. The study was observed that Parsinoplasma of 0.80 frequency occurrence the rate of high compared to the Thalmeta dani. That first species I told these are coral reef associated crabs. The second one I showed the Thalmeta, the species mainly in available on that around 20 to 30 meters, like swimming crabs. The common like the family is opportunity. The outcome of research conclude the two species entirely different mode of feeding methods and based on the feeding component observed in the content analysis. The significance of the refuse and scavengers on the surrounding of coral reef environment through the organism and debris. For my final conclusion is all the crabs they are doing a major role of all the ecosystem, sandy shore and like coral reef environmental and mangrove environmental, seagrass and seaweeds. Those area whatever they like decompose or scavengers materials, those crab they are taking and clean up that ecosystem and environmental also. So we need like a crab importance to the all the ecosystem they doing the major role. So I thank to my ex department Zoology Survey of India. Those work I conducted there only. Those people given a good opportunity to me, and fund provided Ministry of Environmental Forest and Climate Change India. And right now here that this conference mainly I am attend this fund provided Academy of Excellency in Bioscience, and final conclude and thanks to Live DNA and Emerging Scientist Project ACA. Thank you.
Thank you, Dr. Kumaralingam. So any questions to Dr. Kumaralingam? If you have the questions, you can drop in the chat box. So if you don't have, so we proceed the next presenter. I would like to call